ma'am.
1 minute. Good evening, sir.
Good evening, ma'am. Nice to meet you.
Sir, you can start now, sir.
Yes, ma'am. Thank you, ma'am.
I made you a host, sir. Yes, sir.
Okay, okay. Good evening, my dear students. Last class, what are the things already we discussed already, you know about? Here I will recall all those points. What are the things already we discussed in previous classes? See that tissue cultures, already we saw it. What by totipotency? Totipotency is nothing but the any plant cell, it can grow as an entire plant. It has the ability. See that the capacity of generate a whole plant from any cell is called as totipotency. That things also already we discussed. This was first introduced by the name of the scientist called as Haberland. Tissue culture already available, isn't it, my dear? And plant tissue culture is very, very important one, because in within short period we will get it more number of plantlets from in vitro condition. That is nothing but the laboratory condition. That things also already we discussed. And then take an examples of carrot. One experiment was done by the scientists already in our about. The carrot is also cut into the small pieces. Again, it will be split into single pieces. And then again, it will after the splicing, what happened? It will be inoculated into the culture medium. The culture medium, it contained agar and phytohormones, and carbon, nitrogen sources, and other minerals also present in it. These are all are mixed together, and particular pH as well as the temperatures also maintained. Certainly, it will be very effective. Okay, so that medium, what we are going to do now, so the inoculation of the piece of the plant body, whether it may be root tip, shoot tip, or flowers or anthers or whatever it may be, we are placing inside. Okay, so after that a particular period, it may be 1 week or 24 hours or 48 hours, the incubation period is there. After that, what happened? The single cell divided into double, tetra, and octa, and 16, 32, like that, 64, like that. Because it absorbs the nutrition from the agar medium, it also undergoes the mitotic cell division, because already you know about each and every cell, meristematic cell, undergoes mitotic cell division, isn't it, my dear? So that things also happen. After that, what happened? No, automatically it will be produced during the introduction of the axin and cytokinin. Axin and cytokinin, they all are very, very essential for the shoot formation and root formation, simply called as morphogenesis. After that, you will get the plant look like a parent. So once you receive the plantlets in the culture, what happened? The root formation takes place within the test tubes. It can be replaced into where? In the land. That condition is called as already been our about. What is that? Hardening. That thing's also already discussed, isn't it, my dear? Okay. See this one. These are the methods already we saw it. See, this is a carrot. So mature carrot we have taken as a sample. Then you have to cut into a transverse section of the root. That is called as a, so okay, it is also one of the root modification. So that after that, it will also split into some other species, like as a small species like this. After that, the coconut milk culture medium. This is considered as a coconut milk after grinding. Instead of agar medium, they have taken as a coconut milk for their growth. Isn't it, my dear? Okay, you have to place it inside. So what happened in automatic proliferation of cell masses? That is nothing but the callus formation takes place there. That means the single cell or single part of cell. Okay, it may be undergo the multiplication, it uptake of nutrition from the agar medium, or here which what they have used coconut milk culture medium they were used. So used this medium and also multiply like I said. This one. This is complex. More than one tissues united together form of a group of cell. That group of cell is called as what is the callus. After that, what happened? The free cell from the callus continue to develop into suspension. After the development of the mass of cell, it is also split into single cell and they also grow into a suspension. What is the meaning of suspension? Suspension is nothing but the liquid agar medium. Take an example, liquid agar medium. Okay, media. So liquid agar medium we have to take. After that, what happens? It will be embryonic plant and transfer to the again subculture. Okay, subculture. So during this periods, what happened? Automatically. See here. See in this part, the embryonic plant is also further grow into a agar medium, isn't it? The transplant of agar medium culture. Okay. Here, what happened? The single cell, the embryonic plant, it is nothing but the seed like habitat. Don't forget, my dear. Seed habitat. It will grow on agar medium. What happened automatically? This process is called as morphogenesis. Okay, morphogenesis. Morphogenesis is nothing but the formation of shoot and roots. Okay, my dear. So finally, what happened? Undergo the hardening process after the complete development of these two things. So do shoot system as well as the root system. It will be transferred to the land. Okay. After the transformation, what happened? The plant will be grow as a parent. So you will get. So the cycle it will be repeated again and again. This is one of the most important technique. Tissue culture. Okay. So otherwise called as somatic hybridization, micropropagation. So all the three names is here. Okay. We may call see this one. So already we discussed media composition, MS medium, isn't it, my dear? So MS medium already we discussed it. Okay, that things also we saw it, and then we will enter into the remaining part. See that MS medium is very, very important. Murasic and Hook medium. This is very, very important. Okay, so already we saw it in 1962. It is a very widely used different culture system and generally consists of what are the component already we discussed, carbon sources, vitamin sources, organic nitrogen, and growth regulators, and variety of extract, and also gelling agent is also there. Gelling agent is very, very important. Agar. Excuse me. So NEET point of view, tissue culture, which one of the following substances used as a gelling agent? Which one is the gelling agent? Agar is the gelling agent. Okay. So here already carbon sources, particularly two carbon sources, otherwise called as sugar, saccharides, or a carbohydrate, sucrose and glucose. Vitamins B1, nicotine B5, and also B6. Amino acids, very, very important. Two things are the glutamine and asparagine. These are the two important things. This is also very, very important. Mostly important questions. Growth regulator already in our blood. It is nothing but the phytohormones. Phytohormones, isn't it, my dears? Hormones. 2,4-D. Cytokinin, cytokinin. Kinin is nothing but the nucleus. Cyto means cell division, cell dividing hormone. Cell dividing hormone. Don't forget, my dear. Okay. And then the BAP, gelatin. It is also one of the cytokinin component. Gelatin, geamase. It is also obtained from the one of the plant is called as geamase. Geamase. M-A-Y-E-S. Maize. Okay, my dears. So this is one of the plant. Tamil we may call as makkacholam. Okay, my dear. Gibberellin. Gibberellin is also very, very important, my dear. The variety of extract, namely protein hydrolysate and yeast extract, malt extract, coconut milk, orange, and tomato. These are the important points, my dear. It can be kept under which degree Celsius. That is very, very important. 24 degrees Celsius. The pH is also very, very important. Last class we saw it, these things. Okay. then only the growth, it will be very rapid in nature. You know about that room temperature, how many degrees Celsius? 37 degrees Celsius. Okay, our body temperature is also 37 degrees Celsius. Just imagine, both are same. But here you have to place it 24 degrees Celsius. That is also very, very important. 5 to 6 pH. This is also called as acid pH. Okay, the medium should be acid pH. Sometimes they may ask the questions. The agar medium is also tissue culture in agar medium. Which of the following pH it can be maintained? You should maintain like that. They may ask the question, my dear. Okay. Next, we are passing to the next one is called as what is the sterilization technique. What is by sterilization? Already we discussed this part. Okay, in roughly sterilization. So you know the difference between sterilization and pasteurization. Okay, pasteurization. Okay. Pasteurization. What is the meaning of pasteurization? ER. Okay. Pasteurization. Suppose, for example, milk packets, isn't it? They mention milk, pasteurized milk. Okay. Pasteurized milk. Isn't it, my dear? What is the meaning of pasteurized milk? That is nothing but the killing of microbes, particularly bacteria and so on, so spores. Okay, particularly killing of microbes. Okay, how many temperature? Maximum 65 to 75 degrees Celsius for 20 to 30 minutes, 20 minutes. Okay, so that time what happened? Completely killing of bacteria. Okay, so that is very, very important. This is one of the process. What is by sterilization? Sterilization, I think, but one of the procedure. Okay, but here what they are, the elimination of the microorganism, complete removal of the microorganism from the particular place or particular equipments or whatever it may be. Okay, how they will be maintained? How they will be sterilized? Okay, for example, if you want to sterilize any material, you can put into the oven, or particular time duration is also there. Very, very important. Then only aseptic condition it will be obtained. Aseptic means that there is no any microorganism present in the surrounding. That condition is called as aseptic condition. Okay, suppose in our report, some of the microorganism presence everywhere, soil, air, water, whatever it may be, isn't it, my dear? Suppose after sterilization, some of the microorganism, they will be enter, means what happening? It will be infect the tissue culture and also transport the plant, newly discovered plants also, newly inverted, newly produced plants also. So sterilization technique is very, very important. Completely removal of the microorganisms. Sometimes they may ask the question in this point. NEET. Okay. See this one, how it will be. So elimination, complete elimination of the microorganism. For example, take an example. This is considered a small test tubes, isn't it? How will you remove the microorganism within the test tubes? You have to wash with the, which one, alcohol. Okay, H2, O2, hydrogen peroxide. So many chemicals also they were used. Suppose if it is not here, what happened? You have to wash with the soap water. After that, what happened? You have to insert the cotton blood like this after the washing process and roll with the newspaper. Then it kept under the, which one, oven. Okay, it is kept inside the oven. How many temperature within the oven? Here you have to maintain 121 degrees Celsius for 30 minutes. Okay, 30 minutes. So after that, what happened? It will be completely eradicated, eliminated. There is no even single micro, even virus is also killed up to this temperature. Virus also killed under this temperature, 21 degrees. This is very, very important. Here the sterilization is also done. How many temperature is maintained here? 121 degree Celsius. How many minutes? It may be 20 to 30 minutes. That is very, very important, my dear. Okay. So, what about the cooker? Inside temperature is this much. Cooker. Okay. See that one. The maintenance of the aseptic free from all microorganisms or sterile condition is essential for the successful tissue culture procedure. The need for asepsis means what is it? Aseptic. Require that all culture vessels also. Vessels means what are the equipments we are using. Take an examples. What are the equipments we will use or we are using? Just imagine. Okay. Already I told you there is a instrument is there. Okay. Airflow chamber, laminar airflow chamber is there. Okay. This is considered as airflow chamber. Inside there is a test tube is there, tube light. This is called as UV light. Okay, UV. So tube light is also there. This one is called as UV tube light. Okay, my dear. Here, what are the things, first of all, you should note it down. There is a stand is there. Okay. The stand, it contain, what is that? Test tubes. Okay, my dear. So test tubes, how many test tubes you need? At 10 ml or 15 ml test tube is there. Okay. And also one spirit lamp is also present in one spirit lamp. Okay. One spirit lamp is also present. Followed by there is a one small needle like this. Okay, for the sterilization process. Suppose we are using microorganism, they have used this one. And also culture. This is the original culture. Okay. It may be present in the test tubes. Okay. This is the culture. What we are going to inoculate. Mother culture. They told which one mother culture. Okay. And also this one is also matchbox. And also gloves, spirit. Here there is a small bottle is there. That bottle contain spirit for washing process. Okay, this is called as needle. This is flame. This is test tube. Okay, this one is called as mother culture. Sometimes they may be used as a suspension culture. Suspension culture, liquid medium is there. Liquid medium maximum, it may be present in the conical flask. Okay, so like this, all are covered with the cotton black. Okay, all are covered with the cotton black. If there is a medium is the culture. Okay, what are the microorganisms, which culture we are going to introduce or grow. That things also present inside. Suppose take an example, this is considered a plant. Okay. This is a agar medium. So what you have to do, first of all, sterilization is nothing but the sterilization we are doing with the help of the which one, autoclave or oven. Okay. After that, you have to take a sterilized test tubes. Okay. Here you have to fill with the which one, agar medium. Here inside there is a nutrition is there. Nutrition. Okay. The nutrient medium is also present in it. Okay, media. Yeah, the nutrition. I have to draw here. So fully the test tube will be like this. Okay, here the medium is completely, what is that? After solidification, there are some processes also there. This is the test tubes. These are the, this is the way. This is the way. The test tubes look like this one. They make it as like this. After that, it may be placed here, cotton black. Inside is there. Okay, now the plant is there already. Here the mother culture is also there. You have to sterilize this one first, this one. All are doing near the lamp only. We have to do the near lamp, near the lamp only. Before that one, you have to switch on the UV tube light. So what happened? Complete eradication of the microorganisms present the airflow chamber. After that, you have to insert your finger. Okay. And then sterile condition you are doing. Which one? You have to take a needle and you have to sew it on the flame. After that, what happened? A few minutes, what happened? It will be look like a red color. After that, you have to keep it for few seconds. So what happened? The heat will be completely lost. After that, you have to dip into that mother culture and placed into the, which one, new test tubes. All are you have to do within the lamp, near the lamp only. Okay, my dear. So this is after that one, you have to kept it for incubation period. Okay, this is the method. Okay, so that is the important one. See here. See here. See that asepsis required that all culture vessels, media and instrument used handling tissues as well as the explants itself be sterilized. Already I told you. How will you sterilize the explant? Explant means what is the explant means just we have to take, and which one before we have to take which one? This is considered as a explant, isn't it, my dear? This is called as explant. Okay, this is called as explant, isn't it? The explant, how will you sterilize? Because suppose if it has some disease, it will also transfer to all the cultures also. That's why you have to sterilize the explants with the help of HgCl2, mercury chloride, alcohol, okay, and hydrogen peroxide, H2O2. So these are the chemicals that were used for the outer sterilization. After that, you have to make a small cut. That cut is like this. Then again, it will be split into some other things. So that is a process. Okay. Before, how it will be? The explant itself be sterilized. All operations are carried out in laminar airflow sterile cabinet. That's why I told you. That's why I show you the airflow cabinet chamber. Isn't it, my dear? Okay. Go to the next one. See this one. It is essential that the explants, culture vessels, media and instruments used for plant tissue culture be made free from microbes. What is the main purpose of sterilization? What is the main purpose of the sterilization? Free from microbes, whether it may be a plant tissues or whatever it may be. The explants are therefore treated with the specific antimicrobial chemical. That's the things I already told you. So what are the antimicrobial chemicals that were used for, first of all, flame sterilization method used for the sterilization of instruments. Okay, I told there is a small needle. Okay, with a small handle also there. Okay, the needle, what happens, small copper wire is there. You have to kept in the few minutes, kept in few minutes in the, which one, flame. Okay, media. So you have to sew it like this. Okay, sew it like this. What happened? Completely turn into red. That condition is called as what is we are doing sterilization process. Okay. After that, we kept it here. Then it is for only the instruments. Suppose we will not kept which one we cannot be able to get the test tube in the flame. What happened? It burst, isn't it, my dear? So that's why we are kept into the water bath. Suppose take an example. Take a small water bath. Okay. The temperature also here. Okay. Already know about it. Kept into the on the which one whether whatever it may be, stove or electric stove or whatever it may be. What happen automatically? The water bath is boiling. Here inside, what happen? You have to place the stand. The stand inside, what happened? There is a test tube is there. You have to place it like that. These are safety measures. Okay. After that, what happened? You have to remove it and then use it. That's all. Okay, media. Not only that, and also one more is that are continuously used during manipulation work. And then instruments are soaked with 70 to 80% of the alcohol. Alcohol is also one of the important, what is the antimicrobial chemicals. How many percentage they were used? NEET. Don't forget, my dear. How many percentage? 70 to 80. What is the meaning of 70 to 80%? 70% alcohol means remaining 30% water. Okay, that is very, very important, my dear. So sometimes they may ask the questions, 70% alcohol means how much amount of water is present in it. Like that, they may ask the question. Alcohol followed by the flaming on burner in the laminar airflow chamber. Burner in the laminar airflow chamber. Okay, my dears, these are the important sterilization process. This is considered a laminar airflow chamber. See here, my dear. Okay, here there is a opening. Okay, there is a heel is there. Just to take up, you will get the opening. So inside there is a lamp is there. That lamp is called as, what is that? UV lamp. What is that? This is called as, here the UV lamp is there. Okay, UV lamp. UV lamp, what they will do, you know, the entire surrounding, it will be clean now. You have to put before the experiment start. 5 to 10 minutes, just to switch on the UV lamp. What happened? Completely, what happened? You know, this area completely, what is that? Eradicated. The sterilization process also done, isn't it? After that, you switch off the, lamp. So here, what are the things already they are having? See that this is considered as a lamp. Okay, the lamp is there. So here there is a culture. Just imagine. This is a test tubes. Okay, just imagine this is considered as a test tubes. Here the agar medium is filled, isn't it, my dear? And one more is the, here is the mother culture is also there. Mother culture here. This is also a mother culture. This is the new test tubes. Take an example, new test tubes after the sterilization process done. Okay, we have to take from this one. Then you are going to, which one, introduce the new plant. How it will be? See here. So this is considered a mother plant. Okay, here the mother plant is there. In front of this one, you have to take the test tube. In front of this one, there is a flame is there, isn't it, my dear? There is a flame. Okay, you have to do the first one, second one. First test tubes we have to take. Okay, inside, what happened now? From this one, you have to take the X plant and replace to here. Under the, near the lamp only. So after that, you have to do the next one, third one, fourth one. How many test tubes you want to do? You have to do it. Okay, media. So this is the laminar airflow chamber. Sometimes they may ask the question, tissue culture method, which instruments they were used or which chamber we are using? Laminar airflow chamber. What is the meaning of laminar airflow chamber? That is very, very important, my dear students. Okay. Laminar airflow chamber. So after that, what happened? You have to kept under the incubation period. Suppose after finishing this work, complete this work near the lamp, you place it here, and also again you have to sterilize it. Okay, again it you have to sterilize it. Okay, after that, what happened? Kept for few weeks, few days, or 24 hours, or 48 hours, or 3 days, 4 days, like that. You have to imagine. Okay. So after the 3 days, 4 days, what happened? This multiplication takes place because inside there is a agar medium is there. The agar medium was utilized by the plants and also it's produced a mass of cells. That mass of cells. Okay, my dear. The mass of cells. After that, it undergoes a subculture. Okay, my dear. Subculture. Okay. How it will be subculture? From this one, you have to take a petri dish. There are also some, what is that? The agar medium is also poured inside it. Okay, you have to transfer the culture, first culture to subculture, this one. Okay, during this period, what happened? The callus is grow a plant. Okay, media. This is very, very important. Grows a plant. So after that, it may be transferred to the land. After that, it may be replaced into the land. That is called as coordinate. Already so many times I have told you. So these are the important technique. These are the technique they are doing. These are the steps they are doing. This is considered as a laminar airflow chamber. See this technique. What are the things they are doing? Inside only. Sometimes they were used as a which one? There is a pipette. Use and throw pipette is there. There is a small needle-like structure is also mostly they were used for the micro pipette. This is considered as a micro pipette used for the diagnostic process. Take an example: AIDS sample, COVID sample, like that. Okay, they are using, that time they were used, which one, So that this is considered a micro pipette, micro P-I-P-P-E-T, pipette. Okay, my dears, micro pipette. So this is only used for the diagnostic purpose, diagnostic. Okay, diagnostic. Okay, my dear, diagnostic purpose they were used. Okay. So mostly used for the diagnosis. See the safety measures also. They were used the mask also, and also the gloves, all those things. Then it will be shape. Otherwise, what happened? It will be spread throughout the COVID is also spread like this. Okay, media. See here. Most nutrient media. Okay. So what is that? Most nutrient media. What is that? First of all, you should note it down. Media are sterilized. Media sterilization. First of all, you should know. First of all, instrument sterilization. Isn't it, made? First step, instrument sterilization. Isn't it? Instrument sterilization. Second one is explant sterilization. Explant sterilization. Third one is, what is that? Nutrient medium. Okay. Sterilization. So these are the important process. All are sterile conditions only. See this one. The most nutrient media, say most nutrient media sterilized by using an autoclave. I already told you, autoclave is nothing but a small instrument. Otherwise called as it's nothing but the big size of cooker. Okay, what is that? Big size of cooker. Okay, maximum it may be 25 liter. Big size of cooker model. Okay, the size it will be very large in nature, very large in size. We can place the inside, which how many one, here inside we can place it 25 test tubes, 5 conical flask. Okay, likewise. So it can, and also 5 to 10 beakers. So that much capacity it has, isn't it, my dear? Okay, autoclave. The standard conditions for the autoclaving media are 121 degrees Celsius with the pressure of 1 Pascal for 20 minutes. How many minutes? I have to take an example. These two is very, very important. 121 degrees Celsius for 20 minutes. So this is the sterilization temperature. Don't forget, my dear, this is also one of the NEET questions. During tissue culture, the autoclave, it contains which temperature? This one. Don't forget. Plant material can be surface sterilized. The plant material only can be surface sterilized by a variety of chemicals that involve the eradication of microorganisms. Already told you. So many chemicals also already told you. Particularly with the aid of the chemicals such as solutions of sodium hypochlorite. This is the first one. Okay, what is that? This is the first one. And 70% of alcohol. And third one is hydrogen peroxide, H2O2. Okay, etc. They have. And also mercury chloride, HgCl2. So these are the substances that were used for the eradication of microbes. Where? Plant material. Suppose we are using other chemical substance, what happens? It can be destroy the celluloses. That's why they were used this part only, this composition, this chemical substance, this chemicals only they were used for the surface sterilization. Okay, so then only the inoculation or explant, it will be what happened, pure in condition. Suppose if it is disease, mean what is the use of that one? Entire process it will be suffer, isn't it, my dear? There is a reason. Okay. The next one is callus culture. Already we discussed. This is the one of the important method. Already we discussed the callus culture. So callus means what is that? The explants when it undergoes the growth or mitotic cell division started within the test tubes, what happened? It produces a mass of cells, undifferentiated cells, uniform cells, homogeneous cells produced. That cell is called as, what is it, callus. See here. So this is consists a test tube, isn't it, my dear? Here, test tube is that. Just imagine we inoculate all those things. We place inside, there is a cell. We place the inside, there is a small cell, is that. Just imagine. Okay, in this place. See here. So this one. Okay, we place it here. A small cell is there. After few minutes, few days, what happened? No, the single cell, okay, undergo the cell division process. Okay, 2 to 4, 4 to 8, like this. It will be divided. Somatic hybridization takes place, or cell division. Okay, media. After the cell division, what happened? No, it produces mass of cells. Okay, it produces a mass of cells, uniform cells, homogeneous cells. This is also called as a homogeneous, undifferentiated. Undifferentiated means we cannot able to differentiate which one is newly formed, which one is old one. Okay, undifferentiated. That is very, very well. Okay, undifferentiated. Okay, my dear. Undifferentiated cells are produced. This is called as callus. This is the mass of cells. That's why we get there. In callus culture, cell division in the explant, the cell division of the explant lead to the formation of callus mass, which is unorganized mass of cells, otherwise called as tumor cells, just like that one. So this is the plant cell. Okay. Suppose if it is animal cell, just imagine animal cell. Okay, just imagine there is a small single cell animal cell is there. How the cancer cell is also formed. Already you know about it. During the cell division, mitotic cell division, the single cell is also divided into 2 to 4, isn't it? So the spindle formation, all those things already you know about this. This is called as, what is that? Tumor cells, a mass of cells, a group of cells united together form as a tumor. Okay, the cell in callus or the parenchymatous nature. Parenchymatous. This cell is, what is that? Sometimes they may ask the questions. Okay, the callus is also one of the which type of cells? It is a parenchymatous cells. Why it is parenchymatous cell? Parenchymatous cells undergo continuous cell division. That's a reason. Okay, and also meristematic. Meristematic is also the continuous cell division takes place. The callus of the parenchymatous nature. When an explant is placed on the medium, many of the cells become meristematic and they become to divide. That is very, very important. So this is the question, my dear. This is the question. Take an example, 2019. Okay, and also 17. They asked the question, temperature once, and also which type of cells, meristematic, and also sclerotic cells, and also epithelial cell, like that they may ask the question. Sometimes take an example. Plant means what is an explant means what is the nature of the tissues? Meristematic. It is made up of parenchyma. Parenchyma is nothing but the continuous cell divisions. Okay, this is also very, very important. And then temperature, you should note it down. This is very, very important, my dear. This is also very, very important. 22 degrees Celsius to 28 degrees Celsius. This is for the incubation period. Okay, how many weeks we have to place it? 2 to 3 weeks a callus mass is attained. How many weeks it will take means after the inoculation. After the inoculation, take an example here, the explant. Just imagine. This is considered a explant, isn't it? Just imagine. The explant you are placing here, isn't it? After that, it may undergo the, which one, incubation period, which temperature we have to maintain, the test is 22 degrees Celsius to 28 degrees Celsius. How many days we have to? 2 to 3 weeks. Okay. So 2 to 3 weeks means what is that? 15 days. Just imagine, for example, 15 or 21 days, isn't it? 3 weeks. So after that, what happens? The cell divisions automatically occurs within the, we can see it within the test tubes. So it will produce mass of cell. That condition is called as, what is that? Callus mass obtained. With the passage of times, the following three things happen in all type of plant tissue culture. During this time, what are the things? What are the physiological changes which occurs within the test tube? There are three things have been reported. Okay, my dears. See one by one. See what are the things is there. Cell tissue dry matter. Biomass is also increased. For example, you know well about biomass is increases. What is the meaning of biomass? You placed only one cell, isn't it? You placed inside test tubes, which is, what is that? Only one cell is there, plant cell. Otherwise called as explant cell, isn't it? Explant cell, we placed it inside, only one cell is there. After few weeks, 3 weeks, after the 3 weeks, what happened, you know? The test tubes, it contain, what is that? The cell biomass is increased. So here cell division takes place. So single cell into a mass of cell is produced. So this is the first step. This is called as callus. Okay, this is dry matter. Okay, matter, dry matter, biomass. Produced, isn't it, my dear? Increased number. Here one is there, here number is increased. Okay. And second one is, what is that, you know? The level of nutrient in the medium is decreased. Automatically what happened? And the number of cell increases mean here the nutritive medium is this much is there. But here the nutritive medium is this much. Imagine. The nutritive medium, okay? Nutritive medium is decreased. What are the changes occurs within the test tubes? Isn't it? Medium is decreased. This one is called as increased. And then third one is, which one, you know? Third one. Okay. The volume of medium decline due to evaporation. The volume of medium. Sometimes the volume of, what is that? Why it happened? It is a decline due to the evaporation is also another factors. Gradual loss of water, completely the loss of water also. So the evaporation is also one more is there. And gradual loss of water. Water also, suppose the water molecule present in it, it's also lost. Accumulation of metabolic toxin. So inside what happened, you know, for example, the bottom, okay? The bottom what happened, you know? The accumulation, which one, you know? Which one, you know? Accumulation of metabolic toxin. So during the cell division, what are the toxic materials secreted by the plant? It can be stored in the bottom, deposited in the bottom. Okay. The upper portion, what they are having? Only the callus only present in it. Okay. Callus only. Okay, my dear. So this is very, very important. So what are the changes which occur within the cell and tissue dry matter biomass increases. The level of nutrition in the medium decreases. The volume of medium decline due to the evaporation, gradual loss of water, and accumulation of metabolic toxin. Okay, my dear. So for the next one, see here. So this is the explant. Okay, my dear. So this is the explant, first step. So here the nutrient is there. After that period, what happened? The explant is also grow into the callus, isn't it? Callus formation like this. These are the first one. See here. So there are 3 plants they have taken for an example. This is the first sample. This one is the second sample. This one is the third sample. Okay. S1, S2, S3, like that. You have to imagine sample 1, sample 2, sample 3. So here you have to take a first one. Here, this is the explant. After the incubation, there is a callus formation takes place. After the callus formation, subculture is also done. After the subculture, it may be grow as a second. Okay. The same process also done here. Here also samples 2 taken, placed inside the agar medium. The callus formation takes place. The shoot cell biosynthesis, also root biosynthesis also takes place within the test tubes. Third one, same process. See here. This one is also done. This one is also what is the hardening process. See that lab to soil. This one is called a soil process. Okay, my dear. So here, starting from this one to this one. So suppose if it is take like this only. A is to L, up to A to L. See, this is the first one, the second step, third step, fourth step, fourth one. What happened automatically? It will be grows here to here. Okay. So the root and shoot formation takes place. After the roots and shoot formation is also formed to parting process and also transfer to the part. So these are the technique. So this is within short period we are getting. That is very, very important. Within short period means what happened? 15 days you will get the new plant from a single plant. Let's plant cell. Single plant cell. What is that? Single plant cell. What is that? Single plant cell. It gives a new plant similar to that of the parent within short period. This methodology is called as what is a tissue culture. Mostly they were used seedless plant, completely eradicated, completely nil the productions, all those things in the world. Okay, that plants also we can regenerate. That is very, very important. See that another example has given here. So this is the first step. So explant we have to take in and also inoculated. All are with aseptic nature. Aseptic. Don't forget, my dear students, aseptic nature. This is the agar medium. See the how the pure in color. Okay. After that, the callus formation takes place, and shoot system and root system. Finally, this is the last step. Well-grown plant. Well-grown plant. Okay, my dears. So after that, hardening, as it is already in picture showing the shoot formation. See that this is a callus nature. These are the calluses. Okay. The initial stage, this much is the callus formation. See the 1 milligram LP. So like that they have given. Different types of sample have been taken for the callus. Just like a ice cream. Okay, my dear. So these are the different types of callus formation takes place. See here, the mass of cell is 54 is very high. 58 is also very high. These three. Okay. Compared to this one, the last three is very high. In this category, first these two is very high, isn't it, my dear? So like this. And subculturing. Subculturing is also another thing already you know a little bit. It's also another type culture. After the callus has been grown for a while, is associated with the original tissues. It becomes necessary to subculture the callus to a fresh medium. Okay, this is the callus formation. Once the callus formation takes place, it will be transferred to the which one you know. New medium. So after the callus formation, this take an example. This one is called as callus formation. After the callus formation, see here the new medium is taken. Okay. So then only the plant will be grow. Okay. Very rapid nature. That is the things. And growth on the same medium for an extended period will lead to the depletion of the essential nutrition and a gradually desiccation of the gelling agent. That is very, very important. Why we are not growing in the same medium, you know, suppose the depletion of the essential nutrition, lack of nutrition, and also desiccation of the gelling agent. Gelling agent, which one you know, the agar is also reduced because it's all are utilized by the culture. Okay, and then second one, third one is metabolites secreted by the growing callus may accumulate in the toxic level in the medium. Suppose first growing plants, you know, plant tissues, they can secrete some chemical substances that is nothing but the toxic substances. The toxic substances deposited on the agar medium, it will not suitable for the growth of the subculture. That is the third one. That's why we are changing the which one you know, pure medium. Okay, fresh medium, otherwise called as fresh medium, isn't it, my dear? That is very, very important. And then the transferred fragments of the callus must be of a sufficient size to ensure renewed growth on the first medium. That is also very, very important. Size is also very, very important. Sufficient size. Take an example. The cell size is like this, you know. Suppose you are taking this much, it will not grow. Okay, you have to take a proper, which one you know, size also. That is very, very important. If the transformed inoculum is too small, it may be exhibit very slow rate of growth or none at all. That is very, very important. Suppose when you are placing the subculture medium. So the subculture medium is also it contain which one you know, highly nutritive substances and efficient level or more agar. Okay, media. Agar medium is. So, for example, when you are taking the cell is also sufficient size, isn't it, my dear? And also the agar medium is also sufficient. Suppose if it is very less, just take an example. This is considered a large size of callus cell. Okay. The test tubes it contain the nutritive substances. Okay, this much only, no, just imagine this much only. Okay, when you are placing this one, what happen now? The growth, it will be take 1 month or 2 months or 3 months, sometimes maybe. Okay, that's why. Suppose the cell is properly, at the same time, the nutritive substances also this much. So what happened, the growth is very rapid in nature. That is the important. Nutrition is also very sufficient. At the same time, the cell structure is also enough size. Okay, size of the tissue, size is also very, very important. Size as well as the nutritive medium. Okay, so subculture maintain of the pure culture. See here, this is a considered as a mother culture. Already we have taken this one. Okay, from here you are using the needle, isn't it? You have to take the culture from transfer to here to the pure culture. Okay, fresh nutrient present in it. Here we are placing. What happened after a few days or few weeks incubation at the how many temperature? Already know about how many minutes, how many hours. That thing is also very, very important. Okay, 22 to 28 degrees Celsius. How many days per week or 1 week within short period? This plant tissue also grow, isn't it? Okay, in the aseptic conditions. Okay, media. After that one, you can transfer to somewhere else. Okay, media, take care. See the same, the agar medium, callus. Again, here they have given as a subculture. How many times here? They have mentioned here 3 times they have. So this is the first culture. Again, you have to transfer from here to here. The second subculture, here to here. There is a third subculture. This one is very pure in manner. Pure culture. Okay. So this one is a 10% pure means this one is 40%, this one is 95% or 99% pure manner. Okay, the culture it will be transferred from the one test tube to another. The same process also done. Okay, media. See here, how many test tubes is there? Here they have grow in the bottle. All are aseptic nature, different plants also. We can replace the immediately, isn't it? We can replace the immediately, my dear students. Okay, so these are the important points. Don't forget, my dear students. Okay, next we'll pass on to the next one is called as what is that, how they are doing. See this one. Various culture types. You know about the methodology. What are the medium? What are the temperature? What are the pH? What are the chemical composition they were used for the physical? Those things already you know, isn't it? The way culture types was first of all callus culture. Cell suspension. I told you callus means what is it? Is nothing but the single cell. Just imagine there is a single cell into mass of cell. Single cell into mass of. Okay, this is called as callus culture. Cell suspension culture means what is that? You have to take from the cell, single cell, from the callus, single cell from the callus. You have to grow in the liquid medium. Liquid. What is that? Liquid medium. Okay, media, liquid medium. So liquid broth is look like this one. Okay. There is a liquid is there. It contains nutritive substance, all the nutritive substance. What are the nutritive substances essential for the callus culture? All those things present in it. Agar is very less. Okay. You place it inside means what is that? That is tissues. Which one? The callus is also placed inside. Okay. How it will be like this? No. See here. Like this. This is considered as a callus tissue. It can be freely swimming in the, the broth, liquid broth. Okay. So next one is protoplast culture. Protoplast culture already but each and every cell it has a protoplasm. It has a extra chromosomal body, plasmids. Okay, this is also very well. Tissue or organ culture. What are the tissues we can grow? Means shoot tip, apical meristem culture, we can possible. Axillary bolt culture, we can. Root culture, ovary culture, embryo culture, anther and microspore culture. These are the possibilities we can do it. Any plant part already we discussed. What is the meaning of tissue culture? Tissue culture is named any part of the plant body, isn't it, my dear? Here, what are the part we have to take as a test sample? We may take a test sample. Mean, see that's shoot tip or apical meristem. Apical means that. Apical meristem means, just take an example. This is considered as a root. This one is called a shoot system, isn't it? Apical means the tip of the root and shoot. This is called as apical. Axillary. Axillary means but this one is called as axillary, isn't it? Axillary. Root culture. Take an example. Root. This one is root. Okay. Root culture. This one is ovary means but the flower it contains. Okay. Ovary. Isn't it, my dear? Okay. So this one is considered ovary. Embryo. Inside there is a embryo. Is that the embryo culture? Okay. Anther and microbes. Anther and microspore coverage. These are the various culture types we can do within the lab. 8 types of culture have been reported in the media. What are the 8 types of culture? See here, that single cell culture, shoot and root culture, somatic embryo culture, meristematic culture, anther culture, and all haploid production, embryo culture, ovule culture, and ovary culture. All are having in your book. How many cultures? Sometimes they may ask the question, my dear. How many types of culture is the 8 type of culture? Don't forget, my dear students, very, very important. 8 types of culture. 8 types of culture. Okay, don't forget. This is very, very important. 8 types of culture have been identified or introduced. Okay, my dear. So what are the types? Single cell culture. Single cell culture means only one cell. Okay, only one cell. Only one cell. Single cell culture. Shoot and root, already we discussed this. Shoot and root part. Okay, shoot and root. Somatic embryo. Somatic embryo means you can take an examples of ovary. Inside there is an embryo. Is there? Sorry, seed. Seed, there is an embryo. Is there? The embryo can be easily grow. This is also the embryo. Okay. And meristem culture. Take an example. Any part of the plant body. So meristem culture is also possible. Anther culture. About anther. What is that? Anther. Anther culture is there. Okay. So this is also another type. So this one is also anther culture. Okay. Embryo culture, already we discussed it. Suppose take an example, any one of the seeds, the center, what happened? There is a embryo is there, that embryo culture. Ovule culture, this is considered a ovule. What is the meaning of ovule? See here, this is ovary, isn't it? Ovule means you can imagine the structure is look like this one. How it will be? This is ovary, isn't it, my dear? This is ovary culture. Ovule means, see, take an example. Inside there is a female part. Okay, female reproductive organ. There is a 8 nucleated cells also present in it. Here, what happened? Completely covered with this one. So this is considered ovule. Okay, so these are the 8 type of culture have been reported, my dear. Suspensor culture, already told you. The suspensor culture consists of the single cell and small group of cells suspended in the liquid medium. Don't forget, my dear student. So this is very, very important. Liquid medium. Suspensor culture is nothing but which one growth under liquid medium. Okay, my dear. So liquid medium. Usually the medium containing axin and 2,4-D. Axin is also very, very important. Phytohormones, the first hormone, 2,4-D. dichlorophenoxyacetic acid. Suspension culture must be constantly agitated at 100 to 250 RPM. This is very, very important word. They ask the question, what is by RPM? Small r, small p, small m. Revolution per minute, otherwise called as rotation per minute. You have to correctly identify which one is revolution per minute. Okay, revolution per minute, 100 to 250 RPM. Okay, RPM, revolution per minute. Don't forget, my dear student, this is NEET question. RPM, what is the meaning of expansion of RPM? Agitation serves as the following three purposes. Aeration of the culture, that is also very, very important. Constant mixing of the medium, that is also another one. Breakage of cell agitated into small cell groups. Okay, so these are the three important processes also done by the, which one, rotation per minute. For example, what is by revolution means, take an example. Take an example. So you are taking a small test tubes. Okay. Take an example, small test tube. Here there is a medium. Inside what happened? There is a mass of cell is there. Callus. Okay. It is also aseptic condition. This is callus. This is medium. Isn't it medium? Agar. Agar, medium. So we want to separate this molecule into simple molecule. How will you separate? Means you have to take a liquid suspension medium test tubes. Likewise, you have to take 1 to 2. Okay. Okay, like that. You have to take. Here, what happened? There is a liquid is there. Liquid suspension is there. Okay. Here, there is a, what is that? We are transferring here to here. They explain to new fresh medium. Okay. So here, there is a group of cell is there. Here it's also made up of more than one cell. Okay. These two samples, these two test tubes kept under the centrifugation process with the rotator. Okay, rotator look like this one. Rotator means how is there? No, there is a holding of this one. Okay, it can rotate like this. Okay, you have to place it in both the directions. It can rotate. Okay, so during this rotation, agitated means sometimes what happen? No, it may be shaking like this. And rotator, this is also one of the method. Okay, this is also one of the method. So rotator is also there. Rotator is nothing but a small instruments. Okay, small instruments. Okay, small instrument like this. You have to place it here. Okay, there is a small hole is there. You have to place it the test tube inside. Insert inside it. So what happened? It may be rotate like this. Okay, rotate like this. Rotate like this. Okay, or like this. So as a result, what happened? Already we discussed, there are 4 points have been reported. What are the things? Uniformly distributed aeration. Okay, that is very, very important. Aeration of the culture and constant mixing of the medium and breakage of the cell aggregate into small cell groups. Suspension culture grow much faster than callus culture. That is very, very important because liquid medium, so the suspension is also easily absorbed by the root cause and so on. So that's why the suspension cultures grow much faster than the callus culture. Callus is a solid, isn't it? The callus and suspension culture can be used for the following purpose: achieve cell biomass, regenerate of plantlets. This is also very, very important, my dear student. These 2 points. Okay. Achieve cell biomass. Achieve cell biomass means single cell into group of cells. The group of cell into you will get it double cell. Okay. Like that. And another point is the regeneration of the plantlets. Again, from the callus to we can grow as a single plantlets. These are the 2 steps have been reported, and production of the transgenic plants and isolation of the protoplast. These are the important point, my dear students. These 4 points have been reported. So we will finish it up to this level, my dear students. Remaining things we will see the next classes. Thank you so much for the participation. Bye. Take care and good night, mate. Thank you, ma'am.